Today, we're going on an absolutely incredible journey. We are going to follow a sound wave from the open air around us right down into the microscopic depths of our inner ear, and finally into the electrical signals of our brains. It's such an elegant, beautifully engineered biological process, and I seriously can't wait to explore exactly how you are hearing my voice right now. Let's dive in.
Here's our roadmap for today. We'll kick things off with the physics of sound, then see how our bodies catch the wave. Check out our built-in mechanical amplifiers, translate those waves into brain waves, and finally, look at how doctors actually test for hearing loss.
Section 1, the physics of sound. Before we can talk about hearing, we've got to lay down what sound actually is out there in the real world. Because, sound isn't just magic, right? It's straight up physics. We're talking about longitudinal waves. They absolutely need a compressible medium to travel through, like the air you're breathing right now. So imagine an oscillation of pressure. It's just a series of compressions where molecules get packed tightly together and rarefactions where they spread out. Basically, these vibrating molecules are literally bumping into each other, creating pressure differences that eventually reach us. It's super helpful to visualize how this works. Think about the height of the wave. That's the amplitude. A taller wave, higher amplitude, which translates to a louder volume in our ears. Then there's frequency, meaning how many vibrations happen in a single second, which we measure in hertz. A really tight, frequent wave gives you a higher pitch, while a stretched out wave creates a much lower pitch. Now, to measure the intensity of this sound pressure, we use the decibel scale, famously named after Alexander Graham Bell, and it starts right at 0. But here's the catch. 0 decibels does not mean there's zero sound energy out there. Nope. 0 decibels at 2000 hertz is just the absolute minimum threshold of human hearing. It's a tiny reference pressure of just 20 micropascals. To put that in perspective, that is basically the equivalent of a pin dropping in a perfectly silent room. Wild, right? And this comparison is just fantastic. It shows exactly how human hearing stacks up against the rest of the animal kingdom. We can hear from 20 hertz up to 20,000 hertz, with normal speech sitting comfortably in the middle, around 1 to 4,000. But man, the extremes are where it gets crazy. Elephants and moles, they pick up deep, rumbling infrasounds below 20 hertz. Meanwhile, cats and dogs hear all the way up to 40,000 hertz, and bats and dolphins are out there using ultrasounds hitting a staggering 160,000 hertz. Moving on to Section 2, catching the wave. So we know what a sound wave is, but how does our anatomy actually grab it? It all starts with the outer ear, that fleshy, cartilagey appendage on the side of your head. That's the auricle, or pinna. Think of it like a biological satellite dish. It helps localize exactly where a sound is coming from and funnels those waves right down the ear canal. And the canal itself, which, yes, is lined with glands making earwax, actually acts as a resonator. It gives the incoming acoustic signal a nice little boost of a few decibels, just before it smacks into the eardrum, or tympanic membrane. And the eardrum is really where the magic starts happening. It's a vital acoustic transducer, translating those air pressure waves straight into mechanical motion. A normal, healthy eardrum is beautifully translucent and pearly white. But contrast that with an inflamed, red, bulging eardrum. That happens with acute otitis media, an infection where fluid gets trapped behind the membrane. As you can guess, that severely dampens its ability to vibrate, throwing a massive wrench into how you conduct sound. Section 3, the mechanical amplifiers. Assuming the eardrum is healthy, we plunge deeper into an air-filled cavity where sound waves make a huge shift into pure mechanical motion. Let's break down this incredibly precise chain reaction happening with the ossicles, literally the 3 tiniest bones in your entire body. Attached directly to the eardrum is the handle of the malleus, or the hammer. So when the eardrum pushes in, it pulls the hammer with it. Tied together by ligaments, the incus, or anvil, moves right along with the hammer. The anvil then pushes on the stapes, the stirrup, and finally, the footplate of that stirrup shoves directly into the oval window of the inner ear. It is a perfect mechanical relay. But you might be wondering, why bother with these little bones at all? Why not just connect the eardrum straight to the inner ear? Well, the inner ear is filled with fluid, and fluid is a lot harder to move than air. This is where the sheer genius of impedance matching comes into play. First, we've got the hydraulic principle. Your eardrum has a massive surface area compared to the tiny little stapes footplate. We're talking a ratio of over 13:1. This acts exactly like a funnel, taking all that sound energy and concentrating it down to a tiny point. Add in the literal lever action of the bones themselves, and boom, you drastically amplify the pressure so it can effectively punch right into that inner fluid. Okay, but what if a sound is just way too loud? Well, we actually have a built-in defense mechanism called the attenuation reflex. Basically, two tiny muscles leap into action to save the day. The tensor tympani pulls the malleus inward, while the stapedius muscle yanks the stapes outward away from the oval window. They literally stiffen up your ear bones to protect your delicate inner cochlea from super loud, low-frequency vibrations. The catch, though, it takes a fraction of a second to kick in, so unfortunately, it won't protect you from a sudden, intense blast like a gunshot. Section 4. Translating waves to brainwaves. We are finally entering the inner ear, where mechanical pushes spark the electrical signals our brains can actually understand. The inner ear is basically this bony labyrinth hidden away in the skull, and we're zeroing in on the organ of hearing itself, the cochlea. When the stapes pushes against the oval window, it sends physical ripples through the paralymph fluid sloshing around inside. Every single time the stapes pushes in, a pressure wave shoots through that fluid. And to relieve all that pressure, a sort of secondary eardrum at the round window just bulges right out. Now, deep inside this fluid-filled maze is the organ of Corti. And this is where things get truly mind-blowing. It's packed with highly sensitive hair cells. As the fluid waves roll through, these tiny hairs get deflected. The outer hair cells literally act as physical amplifiers, using a special motor protein. Once they do their job, the inner hair cells step up as the true sensory receptors. When they move, they translate that mechanical wave into a chemical release, specifically glutamate, which fires off nerve impulses right down your cranial nerve and straight into your brain. So how exactly does this help us tell different pitches apart? Well, a Hungarian biophysicist named George von Bakisi actually won a Nobel Prize for figuring this out back in 1961. He proved the concept of tonotopy. By doing some wild experiments with strobe photography, he found that the cochlea mechanically sorts sound frequencies by location. High pitches create their maximum vibrations right at the base of the cochlea, while low pitches travel all the way up to the apex. This means your brain knows the exact pitch of a sound just based on which specific hair cells along the spiral are firing off. It's like a piano keyboard rolled up inside your head. Section 5. Testing for hearing loss. All right. Now that we're experts on the mechanics, let's take a quick trip to the clinic to see how doctors test this whole system when things go wrong. Doctors actually use vibrating tuning forks to bypass different parts of the ear. In the Rinne test, they place a tuning fork right on the mastoid bone behind your ear to test bone conduction, and then move it into the air next to your ear. Now, a healthy ear will always hear the air conduction louder. If the bone conduction is louder, it points to a physical blockage in the middle ear, what we call a conductive hearing loss. Then there's the Weber test, where the fork goes right on the very top of your head. If the sound is louder in the quote-unquote bad ear, it confirms that conductive blockage. But if the normal ear hears it better, doctors can cleverly deduce that it's a sensory neural issue, meaning those delicate nerves or hair cells in the cochlea we just talked about are likely damaged. When you step back and think about it, from the physics of air molecules colliding to the hydraulic amplification of your tiny ear bones, all the way down to the tonotopic mapping of microscopic hair cells firing off neurotransmitters, hearing is just an awe-inspiring biological symphony. But remember, those tiny outer ear cells? They cannot grow back once they're damaged by excessive noise. So I'll leave you with this. If our ears are perfectly tuned machines translating microscopic waves into the absolute soundtrack of our lives, how are you protecting yours today? Thanks for joining me on this explainer. Keep learning and stay curious.